So here I am on vacation in a sunny tropical paradise. Actually, I am in a parking lot in a gas station in Hollister. So much for vacation, but we will talk about enzymes. And we talked about processing, and I've talked a lot about different types of processing in previous videos. And I've mentioned enzymes and how cooking or irradiating or um, processing with any number of methods can reduce or slow the activity of enzymes. But now we'll talk a little bit more about these special proteins. Remember, they are proteins. And if we look at what everything is made of that is alive, most of it falls into four categories. There are carbohydrates, things like sugars and starches and similar structures that might be longer, like cellulose in plants, carbohydrates. There are fats or lipids, which are all of the fats and oils and fatty acids. And there are proteins, and there are nucleic acids, DNA and RNA. And a lot of people leave out number 4, the nucleic acids, because it's not something we think about as nutrition. And when we're consuming a plant, a steak or whatever, we're thinking proteins, fats, and carbohydrates. But nucleic acids are significant because they're in every single living cell. So out of those 4, one of them is protein. Protein is made of a string of amino acids, or multiple strings of amino acids in many cases. And so these amino acids, they have an amine group. So don't worry about your biochemistry here, but they have nitrogen. They have a little nitrogen group, and that's what makes it an amino acid. Carbohydrates, fats, and proteins are not all that different down at the most basic molecular level. They're easily interchangeable. We can eat carbohydrates and turn them into fats or sugars, or excuse me, fats or proteins as we need. But what distinguishes a protein is this little amine group or this nitrogen group. And then you have all of these amino acids. We need 20 some odd amino acids in our diet in order to put together the proteins that make up our bodies. And we can also modify many of those amino acids and make them ourselves if our body doesn't get enough through direct food consumption. But then what do we do with those amino acids that are made into proteins? Well, the body uses them in two, excuse me, and the body uses them in 2 very important ways. One is structural. You make something, physically make something. So if you look at the cell itself, the outer coating of cells are made with these special recognition or receptor proteins on the cellular membrane. When cells divide, all of the workers doing actual work inside of the cells are proteins. We have muscles, skin, protein. So we can make things with protein and we can do things with protein. And the proteins that do things that work are often called enzymes. So what is an enzyme? It's a protein that does some special kind of work. So the definition we have here is an enzyme is a biological catalyst that promotes and speeds up a chemical reaction without itself being altered in the process. So that's a catalyst. You may have heard of a catalytic converter in your car. Okay, what is that? Well, it's a special device. When your exhaust is coming out, this device in your exhaust system gets really hot because of the hot gases coming out. And it has special catalytic material on these thin plates that get really hot. So when the gases hit these hot plates, the toxic pollution gases react when they hit the catalyst, causing them to react in a way that burns them further, that reduces them to less harmful gases. So your tailpipe, what's coming out the back, is less polluting. The catalytic converter isn't being changed, at least not in the short term. You know, it has a life expectancy of maybe 10 years or 15 years, but it can go through millions and millions of these reactions on those individual plates at the molecular level. So what are these plates doing? They're causing a reaction to occur at a faster speed or a lower energy. It pushes it over the edge without the catalyst itself being altered. The catalyst can keep going. Once a catalyst helps a chemical reaction, the reaction occurs and goes away. The catalyst is still there, and it can help another reaction, and another, and another, and another. And there are many, many different chemical catalysts that we use in manufacturing, that we use in automobiles, that we use in factories that clean up the stacks of waste coming out of a factory to clean it up before it goes into the air. But our bodies are using catalysts all the time. But because it's in our bodies, it's a biological thing. It's one of the major components, a protein. We call those enzymes, specialized biological protein catalysts. And catalysts can do two things. They can cause a reaction to make things to get bind together. They create things. They join things. Those are called anabolic enzymes. Anabolic builds or puts together. Usually, we're talking about the other type, the catabolic enzymes. Catabolic breaks things apart, breaks them down. It's useful in ripening. It's useful in digestion. It's useful in many, many biological processes. So how does an enzyme work? I described it to you briefly, but it lowers the activation energies of a substance. In other words, if I'm talking about a catabolic enzyme, to break it apart, essentially it lowers the energy required to break it apart. You could think of it as putting stress or tension on a bond. And in many cases, that's what we think the enzyme is actually doing. It's putting tension on a bond, making it much easier to break that bond. Or in other cases, it lines up The reactants, if it's an anabolic enzyme, a building enzyme, it lines them up. It just puts them in a place where it's much easier to join them together. We call that the activation energy, the energy required to make the chemical reaction occur. And it can occur at a much lower energy. Think about this, sugar. Is sugar flammable? Yeah, it burns. Doesn't burn really well, and it needs to be hot, but if you put it in a situation where you have enough oxygen and you put a flame to it, sugar burns. Sugar burns with oxygen to create energy, just like wood, paper burning. But there's a problem getting energy from sugar at that temperature. It would kill you. You can't get energy from burning. It's too hot. It will kill you. So what do you do instead? With enzymes, you can burn the sugar with oxygen. You can react the sugar with oxygen in order to break down that sugar molecule in the presence of oxygen, making carbon dioxide and water. But instead of doing it at the high energy of fire, you're doing it at a much lower energy at the temperature inside the body, 98.6 in our case. So we're doing the same chemical reactions at a much lower energy. So if we look at an energy profile, here's the reactant or reactants and the products. And EA is the energy of activation. And some heat energy must go into this reaction to make it occur. So in this case, how much energy does it take to turn this reactant, this molecule, into the products, breaking it apart? Now, if it was sugar, we would need two reactants, the sugar and oxygen, and then it comes out as carbon dioxide and water. Now, there's a couple of steps in that, so that's probably not the best example, but this is maybe just one step in that process. And so you need to put in energy in order to get it over the hump, in order to break that bond. And then once that bond is broken, now you have your products with their own inherent energy. The reactants have their energy, the products have their energy, but it's not a straight line from reactant up to product or down to product, depending on the type of reaction. It's not a straight line. You have to get over this activation energy hump. If a catalyst is present, it lowers the activation energy. So to go from reactant to products, boom, you have a much lower hump to get over. It happens much easier, much quicker, less energy required. And the catalyst is there as a participant, but the catalyst is not directly part of the reaction. The catalyst is unchanged. It participates in the reaction by lowering the activation energy, by either holding the reactant, in this case, holding the reactant in a way that puts tension on the bond, making it easier for that bond to break, or going in the other direction, holding the products, which in this case would be the reactants, if I'm going the other way, holding those reactants, two reactants, in a position that makes creation of that bond much easier. So enzymatic reactions. Enzymes combines, or an enzyme combines with a specific substrate to form an enzyme substrate complex. When we look at enzymes at the molecular level, they act like a lock and key mechanism, where the enzyme is like a lock, and all of its shape as well as its electrostatic charges, positive and negative charges, and hydrogen bonding, all of the pieces of that enzyme line up perfectly with the key, the substrate itself. So we could look at it this way. Here's the enzyme, and with its combination of shape and charges, it can interact with the substrate, and now that substrate forms the enzyme substrate complex. The bond is broken. The products now are released, and the enzyme is unchanged and can go back and do it again and do it again and do it again. Now, there's a life expectancy. It's not like enzymes are eternal. They do need to be replaced. And so the body's not only making new skin cells and new hair cells, or plants are making new leaves, they're also constantly remanufacturing and making new enzymes to replace the ones that are getting old. They're mechanical entities, and they do wear out. At the molecular level, they don't wear out in the same way that we think of gears and things like that, but they can get damaged. So round and round we go. So what about the structure of an enzyme? Now, enzymes, I've told you, are proteins. And that is true, but there also is a non-protein component to enzymes. And that helps give it its final lock and key shape and charges. So there's either a coenzyme, something that works in combination with the enzyme, and in most cases, coenzymes are vitamins. Why do you need vitamins? You've heard about vitamins all the time. Get your vitamins, eat your vitamins, eat those leafy greens and get good vitamins. What the heck are those vitamins actually doing? The vitamins are the final ingredient, the final piece to make enzymes that do everything in our bodies. Now, different enzymes are requiring different coenzymes, different vitamins, or other similar components. And we also have cofactors. Now, don't worry about coenzymes and cofactors, which one is which. I won't ask you that on a test. This isn't a biology class, but you should at least recognize the words coenzyme and cofactor. And you should know that vitamins and minerals are important because of what they do with enzymes. They make enzymes functional. Now, one thing you maybe already know, hemoglobin in our blood has iron. Iron in the blood is in the molecule hemoglobin, and that allows the hemoglobin in our blood to carry oxygen to all of our cells and carry away carbon dioxide as the waste product from our cellular respiration. What is hemoglobin? A massive protein. And it moves oxygen around. It moves carbon dioxide around. It wouldn't function at all without the iron, the mineral in the center of the hemoglobin. It is the iron that gives it its final shape and function. And that is true of all things enzyme. So what impacts enzyme activity? Let's take a very short break and be right back, and we'll talk about this.